Good afternoon, everyone. My name is McCain Vogel. I am one of the associate editors here with No-Till Farmer magazine. So I just want to say thanks for joining us today for the digital demo with Cultivates. This presentation will feature Wayne Sledge. As you can see, he's the founder and president of Cultivates. Wayne has over 50 years of experience in the field of agriculture, and he actually began his career as a grower before eventually transitioning into fertilizer sales. I'm going to let him talk lots more about his background just a minute, but before we get started with the actual presentation, just a couple housekeeping items for you. So on your screen, you'll see on the control panel down on the bottom, there's a Q&A icon. So you can use that to submit questions if you have any throughout the presentation. We're going to hold those until the end. We'll save about 15 or 20 minutes at the end of the presentation, and then we'll try to answer as many of those as we can for you. And then just a reminder, we also are recording today's presentation. So in the event you want to go back and watch it again or share it with anyone else, typically you'll receive that link in your email about 48 hours after the conclusion of the presentation. So without further ado, Wayne, I will hand it over to you.
Great. Thanks a lot. And I'd like to welcome everyone in. Thanks for joining us. And hopefully we can share some information, give you some insight into some of our products and how we perceive foliar applications of nutrients. I guess I'd like to start by talking a little bit about myself and let you know kind of who I am and how I got into all of this and where I came from. When I was a kid, about 13 years old, started working on row crop farms in the Willamette Valley. And basically, back when I was young, all of the crops were hand harvested for the most part. And so when you're 8, 9, 10 years old, you started out hand harvesting crops. And it didn't take me too long to figure out that I wasn't worth a darn at doing that. And if I was going to have any school clothes and didn't go to school looking like a vagrant, I probably was going to have to earn some more money. So started working by the hour, doing things like moving irrigation pipe, bucking hay, those kinds of jobs. When I was 19 years old, 1966, still in college, decided I wanted to take my hand at farming. And so with the help of the family farm, started out farming and raising some row crops on my own. The late 60s were not very good times to try and do what I attempted to do. And unfortunately, I had a really bad habit. I like to eat. So I figured I probably better find a full-time job somewhere. Had the opportunity to get into the fertilizer ag chem industry. And so in the early 1970s, I started out selling fertilizer and ag chemicals, mostly doing sales and consulting. And for the next 30 years, managed multiple fertilizer plants, worked in retail as well as doing some wholesale fertilizer sales. In 2006, founded Cultivates. And when I started, I just had 3 products that I started with, building potassium acetate, calcium acetate, and copper acetate. So that's kind of how I started this whole thing. And I started my own business with 2 key objectives in mind. Our products, number 1, they got to be the highest quality, and 2, they must make the grower money. So if they don't meet those premises, we're not going to make it, we're not going to sell it. The grower has to make money with the products that we sell. Where we are and where we're going, we're on the West Coast. Our facility is located just north and west of Salem, Oregon. And at the present time, we market products on the West Coast, Oregon, Washington, California, Arizona, Nevada, Idaho. And we're making a concerted effort to push into the Midwest through distribution. So we've been working there for the last couple of years, trialing products in the Midwest region and trying to get into that market. So that's where we're headed and where we're going. Products are used on a lot of acres out here on the West Coast. So we're tried and true, but we're pushing to the upper Midwest. Could your yield be better? I think everybody always thinks of that when they're farming. I mean, that's the number one thing you're going to focus on is your yield and maybe what's holding you back. So when we start looking at that, plants may not be getting the right nutrients at the right time and in the right quantity. So that gets to be a real issue is which nutrients can we get into that plant? How can we make the plant do what it wants to do better? And I think that's our primary focus. And I think that's what everybody should think about is trying to get that plant to perform that at its maximum potential that it can do. So that's what we're trying to do. One of the other things we need to look at is how do we get those nutrients into that plant? On a foliar, what are we going to do to help get it in there the most effective, most efficient way that we can? So there's a lot of different carriers out there. Primarily, the most common that I see is EDTAs. And that may not be the most effective way to get that in there. So we're going to take a little look at that and see what that looks like. For most products, nutrient concentration is low. You'll see a lot of products that have 0.1, 0.2%, some even 0.005 that I've seen out there. So pretty low concentrations in some of these foliars that are in complete blends. Why would you choose a foliar? When it's applied at the right time and in the right quantity, they go into a plant really, really rapidly. And one of the things that they found early on in the mid-1950s, when they started doing some radio tracing, one of the things they found was that when they applied a nutrient to the foliage and then followed it, they found that it could move at up to a foot per hour. So you can see that if you're putting a foliar on a plant that's 2 feet tall, in a couple hours, it's already the root system. So it's really a fast way to get something into that plant. Doesn't take long to get it in there and for the plant to be able to utilize that nutrient. It's totally unaffected by soil conditions. Some of the conditions can make nutrients ineffective because there are times that things that you apply to the soil or things that are naturally there, one will interfere with another. So it might be on a soil test, but you might not be able to get it into that plant. And foliars are readily available, gives an easy access to nutrients compared to soil-based fertilizers. So the foliars are pretty quick to get into that plant. If you compare the way that the reaction that happens, if you look to the left, you'll notice a foliar delivery system. When that nutrient is applied to that leaf surface, it can go in through the stomata or across a semi-permeable membrane on the leaf. And if it's the right size, and if it can get in there, the plant can use it very rapidly. Whereas on the right-hand side, if you look at a soil situation, nutrients can get tied up. And the other thing too is that you need to realize that those roots are exploring only a small portion of the soil. So there might be a lot of things in there, maybe for different reasons the plant can't extract them out of that soil. So that's something I think you need to think about with soil applications versus foliar applications. The other thing too about foliars is typically we'll look at the micronutrients because it's an easy delivery system. A good way to get micronutrients into that plant is through the foliage because it doesn't take very much. You can affect a large change with a small amount of nutrient. Advantages of acetates for nutrient delivery. Basically, we build all of our products in an acetate form as far as our micronutrients are concerned. They outperform other carriers such as an EDTA and their ideal delivery for micronutrients. One of the things that you need to think about when you look at the delivery system is the thing that you're putting that in there with, which we're using acetic acid. Acetic acid is a plant part. Plants naturally make acetic acid. And the reason they're making acetic acid is it's one of the organic acids that plants naturally make to use as a root exudate to extract nutrients out of the soil. They're easier for plants to absorb. Acetates are small in size, so it can be readily absorbed through the leaf cuticle and the stomata. So they're really, really small particles. They're easy to break down. They're not a chelate. Or, EDTAs are a chelate. And the word chelate means claw-like. So that's the way that bond is made. An acetate's a complete molecule. So it's a little different than what you would think of as an EDTA, but they're easier to get into the plant. You look at nutrient breakdown and absorption. On the left, you'll see what an acetate looks like. It's very small. It's easily absorbed. The plants accept it as a native substance, so it's not something foreign to the plant. On the right, you'll notice that that's a depiction of a chelate. They're a lot larger, and that can prevent even getting into the plant. And then once it does, the plant has to look at it and say, what is this? If the element does get out of there, the other concern you have is you remember that it's a chelator, it's going to grab something. So what else does it grab in the plant? Does it just leave on its own, or does it take something with it? So that's another consideration that you have with an EDTA. If you look at the size of the molecules, one thing that you'll notice is on the left, you'll see a depiction of an iron acetate molecule. So you notice that there's only 2 bonds that are holding that iron in place. So it's not a very large molecule. It's fairly simple, fairly small. On the other hand, on the right, look at an EDTA iron. Look at the bonds. Look at the way it's held. You've got 6 sites that are holding that iron in there. It cannot get out of there. It's held tight. Another way to look at these is of size is to look at weight. If you look at iron acetate, it's about 174 grams per mole. And EDTA is 435 grams per mole. It's 2.5 times heavier, 2.5 times larger in size. One of the things that I want to talk about a little bit is why would anybody make an EDTA? It's fairly simple. If you have an EDTA, it's held so tightly that it can't interact with other elements. So if we take an iron EDTA and a zinc EDTA and a manganese EDTA, we can throw all 3 of those together and not have an interaction because they won't interact with one another. Well, if that's the case and they can't interact with one another, then how is it they can interact with the plant through the foliage? Just something I want you to think about. So the advantage of having an EDTA is more an advantage for the manufacturer than it is for the grower. They can throw those things together in any combination they want, put whatever they want on the label. The label doesn't say it has to work. The label just says it has to be in there. When we look at rates of absorption, this is a trial that I'm sure most people have seen when they see potassium acetate. This was done at Texas A&M in the early 2000s. And so you'll notice that potassium acetate is 5 times more effective at getting potassium into that plant than any of the other potassium products that you would normally look at. And when I first saw this in the early 2000s, my immediate response was, gee, if I can get potassium in that form more readily, then why can't I get the other elements in the same way? And so that's what led me to develop the line of acetate products that I have, is because I felt that if in fact this is a better way to deliver the elements to the plant, then that's what we should be doing. We should be using that as a delivery mechanism to get those nutrients to the plant. And that led us to the development of what we now call our acetate foliar nutrition. We have a full line of acetate products. They're custom proprietary formulations that we have developed. They're pH adjusted. And what I mean by that, basically, I think most people understand when we're doing a foliar that we want to keep our pH in the range of high fives, low sixes. And so that's kind of what we do. We try to keep those so that actually they will act as a buffer. Typically, we're in the range pH range of around 5.5. They're pure and they're clear, and we try to get a good concentration in there. We put as much nutrient in each gallon as we can possibly put. Some of the advantages: we're science-based. Forget spray and pray. Science-based approach focuses on specific nutrients for the best results. So we're trying to look at timing and put the right nutrient in there when the plant needs it. We're ultra filtered. That means we don't have sediment. We don't want clogging, loss of product during application. So it saves you time, frustration, and money. And I think from the perspective of being a field man, the last phone call that I ever wanted to get was from from a grower, from a grower who said, "You know that stuff you just sold me? My sprayer's plugged up. Why don't you come out here and help me clean it out?" Not a good phone call. We have measurable results. Tissue samples show the increases that we can get with our products. Our formulations typically are going to run from 5 to 8% of nutrients compared to 0.1 in a lot of other ones that you'll find on the market. So we're getting as much nutrient in that product as we possibly can. 1 gram of fertilizer per acre isn't enough. What led me to do this was I sat down one day and asked myself, if I have a formulation with 0.1% of a nutrient, how much am I applying? So I said, okay, I'm going to put on 1 quart per acre of a formulation that's going to weigh an average of 10 pounds per gallon. So how much am I getting? And the math works out to 1 gram per acre. I know a lot of people probably aren't really familiar with metrics and a gram. So let's change it a little bit to put it into focus. There's 28.5 grams in an ounce. So let's take 1/28 of an ounce, I'm sorry, cut that back, divide it up to 43,560 pieces, and put each one of those on a square foot. Got a feeling you're not going to have a whole lot of product per square foot. So you might be thinking about that when you're looking at how much concentration your foliar has. One of the studies that I found doing research a few years ago was a study that was done in Arkansas on EDTAs and their effect in foliar absorption. The thing that they were looking at in this trial was they were looking at the efficacy of using an iron and zinc EDTA versus other forms to apply those nutrients. One of the main findings of this study is this study showed that EDTA should not be used in trace element foliar sprays. Again, remember, this is university work, and their conclusion was don't use an EDTA as a foliar. Soil applied EDTAs are fine. That's where they really are meant to work and where they work the best. But on leaves, they just don't work. That's not a place to be using them. Now let's take a look at some of the trial work that we've done. And this is basically stuff that we have done recently. These are trials that we have conducted this year. When we look through these, I want you to realize that these are not just one application. What we did was we did 4 reps of each product and averaged those 4 reps, complete randomized block design. And they were field size trials. This isn't something that was done with a hand sprayer. And if we look at manganese and look at the untreated, you'll notice that we got about 120 ppm in the leaf. We go to an EDTA, we got 132. In this one, we even tried some amino acid chelate, 134. Essentially, those are probably about the same number. But then look what happens when you use an acetate. The manganese goes to 171, which is almost a 30% increase above the EDTA. Iron is another one we looked at. And so in this case, we also wanted to take a look at an EDTA. And I realized that there's a lot of EDTA used in soil applications, especially where you have calcium-induced iron chlorosis. And EDTA is very effective in that situation, especially in high pH situations where EDTAs are not very effective. But when I looked at labels, I noticed there was a lot of them that were referring to foliar applications. So I thought, well, let's take a look and see what happens. So let's look at an untreated. We see 71 ppm. We put on an EDTA, and look what happened. Our iron went down. However, when we used an acetate, we're at 102. So we got almost a 54% increase above the EDTA. So remember when you're using these that just because it has a chelator in there, it doesn't always mean it's going to work. And so what this shows me is that EDTA holds it even tighter than an EDTA. So that's something I want you to realize when you're using these things. If you use the right product in the right place at the right time, everything will probably work. One other thing we looked at was zinc. This was a trial actually we did last year. So if you'll look at the untreated, we got 31 parts per million. The zinc EDTA, we got 33, which is statistically about the same. But when we used the acetate, we went up to 80 ppm. I'd like to shift gears just a little bit here and take a look at soil and nutrient interactions and some of the things that can prevent things from getting into the plant and why we would have to use foliars in the first place. I don't know if most of you have seen one of these, but this is a Mulder's chart. And when people first look at one of these, I think the immediate reaction is, oh my gosh, what is I'm looking at here? There's circled with arrows and lines and dotted lines and just going every direction. Can I make any sense of it? We're going to break it down a little bit and explore some of the nutrients and interactions. But before we do that, I want you to realize, what it says here. For instance, I want to take a look at N, P, and K, which all of us apply. And I'm not going to say it's wrong to apply N, P, and K, but what I am going to say is when you do that, realize what's happening. For instance, a high N levels can limit your boron, potash, and copper uptake. And it also increases the need for magnesium at the same time. So remember that. High phosphate levels affect iron, calcium, potash, copper, and zinc absorption. High potassium levels can decrease magnesium availability. And also the dotted lines actually mean that when you apply that, you have an increase for the need of that element. So when you look at potash and look at manganese, you'll notice a dotted line there. You put on potash, you need more manganese. That's what that means. Without a balanced nutrient supply, excessive nitrogen, phosphorus, and potassium in fertilizers can cause deficiencies in essential nutrients. Want to take a look a little bit at the different elements. Copper, some of the functions. Now all of these that I'm going to talk about, all of these functions and interactions we got from Marchner's book, The Mineral Nutrition of Higher Plants. So that's basically our reference for all of these. These aren't something that we made up. The functions of copper, it's an activator of plant enzymes, plays a role in protein synthesis, plays a role in photosynthesis, plays a role in respiration, nitrogen metabolism, and it's 100 different proteins in the plant contain copper. Typically, when I think about copper, some of the things I think about are some of the other roles that maybe it might play in the plant. Cell wall lignification is certainly one. So when you lignify those cell walls, you keep the plant more erect. Another thing that it does is it helps the bark to be flexible, like in a tree. So you impart flexibility, so it allows the plant sap to move a lot more easily in the plant. Another thing it does is it works in pollination. So really key in pollination. And hopefully nobody from EPA is listening or anybody like that, but it's also a natural fungicide. So it can do that as well. Some of the things that cause interactions, large nitrogen applications, as you saw, can induce a copper deficiency. Soils high in organic matter can induce it, and calcareous soils have the potential for copper deficiency. And if you look here at copper, when you apply it, it's kind of hidden behind the picture of me, but it will affect manganese and iron. So copper will depress those two elements. That's what this is showing. So that what we've done is we've taken that Mulder's chart, pulled it apart just to show the interaction a little more easily to see. Calcium. Calcium's function is it's used in the construction of cell walls. It's important for cell wall permeability. It's a messenger in the plant, and it's used in conjunction with boron. Actually, to get calcium in, you need boron to be available. And it must be present for formation of new cells. When I think about calcium, the other things I think about is growth energy. It has huge amounts of growth energy for that plant. And I want you guys to always think of it as, just remember that calcium is a fertilizer. It's not something that's in lime that we throw on the ground to change the pH. It's a very, very important nutrient. It's responsible for cell wall division. So if you think about a flowering plant, or any time you have a flower present, from full bloom forward, all of the cells in that fruit are being developed. And so in order to get that cell wall, the cells to divide, you need calcium. Calcium goes into those cells and it does not move. Calcium is not mobile in that plant. So it can't be in the cell, then if the plant needs it somewhere else, like some elements will come out and actually move, like phosphorus or potassium, are very mobile in the plant. Calcium, on the other hand, is not mobile. It does not move. Potassium and magnesium can prevent plant uptake. And you can see here that when you apply calcium, I mean, it goes after almost everybody. So high calcium can depress a whole lot of different elements. Potassium. Potassium's function, it's essential for transportation of sugar and formation of starch. Responsible for opening and closing the plant's stomata. So it's really key in the guard cells around the stomata, making the guard cells, giving them the ability to open and close and help in transpiration. Increases your flower size, quality, and quantity. So if you want bigger flowers, more flowers, which usually equates to seed, and higher quality, you need to get potassium in. It's used in cell extension. When I think of potassium, I guess the word extension is not the word I usually use. I usually use the word expansion. And as it relates to plants and growth, after bloom for the first 20 to 40 days after full bloom is when we're using calcium in that plant to divide those cells. After that, we need to switch our focus to potassium because now we've got the cells, now we need to expand those cells. So we need to switch our focus from calcium to potassium for that cell expansion. 98% of soil potassium is fixed and unavailable. There's an awful lot of potassium in that top foot of soil, but you don't get very much of it. So small amounts is all we actually get to use out of that vast amount of potassium that's in the soil. Plants deficient in potassium will uptake sodium in its place if the sodium is available, which is really not good for plants. Calcium and magnesium can prevent the uptake. And moisture can be a limiting factor in potassium availability. In fact, when that soil gets completely dry, you don't get any potassium. If we look at a potassium application, what you'll notice is that it's going to depress boron. But as you apply potassium, your need for manganese and iron go up. And typically, one of the things that we've just developed is we are now going to start marketing a product that has both potassium and manganese in it. So we can apply both elements at the same time. Magnesium. Magnesium's function is chlorophyll. You have to have magnesium in chlorophyll. Used in enzyme regulation, used in protein synthesis. Sandy soils can be deficient. High soil levels of potassium, calcium, ammonium, and manganese can depress the uptake. And you can see here as you apply the magnesium, it's going to depress your potassium, but increase your need for phosphate. Zinc. Zinc's function, structural component in enzymes, involved in energy metabolism, involved in synthesis of plant protein, controls the synthesis of IAA. And what is IAA? IAA is indole acetic acid. What the heck is that? Indole acetic acid is an auxin. What the heck's an auxin? Well, auxins are maybe a little different, kind of a funny kind of a critter, because most farmers have used synthetic auxins, probably not realizing what they were doing. But anybody that's ever put on 2,4-D has applied a synthetic auxin, because that's exactly what that is. And it sure as heck makes some plants grow themselves to death. The other thing that auxins do, they are produced at the growing point of the plant. They make the plant have apical dominance, which means it wants to grow straight up. They're transmitted down through the plant to the root tips and tell the roots, look, I'm growing like mad. I need more roots because I need more food. So zinc is a very, very important nutrient. Interactions, low organic matter soils and sandy soils are often deficient. Large phosphorus applications can induce zinc deficiency. I think most people are aware of that because most soil applied nutrients that you see that are phosphate containing normally will have zinc with it. High calcium soils can prevent zinc uptake. Photosynthesis in corn is more greatly affected by zinc deficiency than soybeans. Iron. Function of iron is important in the formation of chlorophyll. It serves as an activator for biological processes such as respiration, photosynthesis, and nitrogen fixation. Most soil iron is unavailable to plants. So it's there, but the plants aren't going to get it. They're not very easily. Deficiency is common in alkali or calcareous soils, and that's why we see the situation where we have calcium-induced iron chlorosis. High calcium levels will block that. High levels of zinc, manganese, and copper can prevent uptake. Poor aeration in wet soils can prevent uptake. Corn has a higher iron requirement than soybeans. And there again, if we apply iron, we're going to tend to depress our phosphate. Molybdenum. Molybdenum's function required for nitrogen utilization, used in respiration by nitrogen-fixing biology, can be dangerous for forage animals. Normally, most labels, when you put a certain amount of molybdenum in them, there will be a statement on there that it can be harmful to ruminant animals. So that's what you'll see in there. When I think about molybdenum and in fixing biology, my thought immediately goes to legumes. And so if you don't have the molybdenum available for that biology to use, then you're not going to have very good nodulation. Your in fixing is not going to be very good. And one of the easiest ways to determine that is with your legume, dig up a plant, take a look at your nodules, and then break them open. So when you break open a nodule on a legume and you see white or cream colored inside, probably not getting much utilization. When you break that open, it should be blood red if everything's functioning like it should. So maybe a situation where you don't have enough molybdenum. Interactions, acidic soils can prevent molybdenum uptake. It's different than a lot of the other micronutrients because it works just the opposite way. In acid soils, it's not available, unlike a lot of the other ones that the availability goes up in acid. Sulfate can suppress molybdenum. It's highly mobile in the plant, so foliar application can really correct the deficiency very rapidly. Manganese. Manganese is used in photosynthesis. It's an activator of enzymes, assisting chlorophyll production. The other thing I think about when I think about manganese is that you have to have manganese to have seed. And typically, most of the crops we're raising, we're trying to get seed. So you need to really be keenly aware that your need for manganese may be a lot greater than you think. And so sometimes you can get a response to manganese to seed development because, like I said, no manganese, no seed. Availability decreases over pH of 7. High levels of iron, copper, and zinc can reduce the uptake. High levels of organic matter can prevent manganese uptake. Potassium applications increase the need for manganese. And like I said, that's why we started putting manganese and potassium together, because we noticed a huge increase in yield when we did that in some crops. Glyphosate can induce a manganese deficiency. Just remember the one thing about glyphosate, not only are EDTA chelators, glyphosate is also a key and a really strong one. That's about all that I have on that. I would like to take this opportunity to introduce our team to you. Our team members, Matt Clark. Matt takes care of Pacific Northwest sales. He's also our general manager. And Matt's also responsible for all of our freight incoming and outgoing as well. So he gets to wear a lot of different hats in the company. Doug Elliott. Doug does all of our Southwestern sales, California, Arizona, Utah, Nevada. Those states is what he travels. Chad Calhoun is on the eastern side of Iowa. Chad takes care of most of Iowa, Indiana, Illinois, Ohio, Western Kentucky, and parts of Missouri. So that's his area. Chuck Propel is in Minnesota, in the northern part. Minnesota, Wisconsin, North and South Dakota, part of Iowa. Brock Corsi. Brock does our marketing and advertising. Brock also was really keenly helpful in doing all of our prep work on our trial work the last couple of years in the Midwest. He's been in Chicago for the last few years, and he's going to transition over to the head office this year. So anyway, that is our team members. If you have any questions, get a hold of any of us, and we'll try and help you solve any problems you might have. This is a lineup of our products. So this kind of shows you what we make. Now, one thing I will say about our products is when you see all those colors in there, yeah, those are kind of pretty, but I got to tell you, those are natural. We didn't put any dye in those or do anything to change their color. So that's the way they are. And at this point in time, I guess I would like to thank you for participating. And if you have any questions, open the floor to those questions this time. Thank you.
Yeah, all right. Thanks, Wayne. It looks like we've got a few questions already. And if you missed my announcement at the beginning, there is a Q&A icon at the bottom there. So if you do have a question, we've got about 15 or so minutes to try and go through some of those. So we'll get started. I think this one came in first, Wayne. Question is, how long do the tissue levels stay up? Is it roughly 2 weeks like other Foliar products?
Yeah, I think that's what you're going to see. I mean, you got to realize that any of the foliars are going to be a short-term fix. It's not going to be a long-term fix. So I think if you get a couple of weeks out of them, I think that's what you would expect.
All right, very good. And then next question, can you mix together more than one of the Cultivates products for application, or do they need to be applied separately?
You can tank mix them. The problem you have when you're mixing it in high concentrations, like what we would do for a product to ship to somebody, these products are highly active. So you got to kind of know what you're doing in order to get them to go together. So we know how to mix some of them, but in a tank mix situation, there are no issues.
Then similarly, can your acetates be mixed? Calcium, zinc, and potassium are generally lower testing for this particular respondent, and says sometimes these do not play well together in a mix.
They will go together in a mix. However, you said calcium, potassium, and zinc.
Zinc, yep, that's the yep.
Okay. Okay. Here's one of the things that I probably you will see me never do, and that is apply calcium and potassium at the same time. Those two cations typically will fight one another. So it will take the one that you give it preferentially the highest of. So like I had explained earlier, the calcium needs to go on at cell division, the potassium needs to go on at cell expansion. So I will rotate those two. I will typically never put them together.
All right. And then if there are higher nutrient concentrations in your formulas and higher uptake potential than EDTAs, do you apply at a much lower rate than EDTAs to prevent toxicity?
I don't apply lower rates because the plant actually needs those higher rates to perform like they do to get the amount of nutrient that's in there. Toxicity is not an issue. We're not putting on nearly high enough rates to cause any toxicity of any of the nutrients to any of the plants. I've never seen that. In fact, I was challenged one time a number of years ago about copper, and that I could, with my copper acetate, that I could cause a toxic situation in trees. I happen to have a small cherry orchard. I went out and used a 10x rate without any phytotoxicity. So no, I think our rates are just fine. Toxicity is not something I would be concerned about.
All right. And then this question is, do you have any results on citrus affected with citrus greening disease?
I know what citrus greening is, but no, I have not had any experience with it, so I don't know.
All right. Well, that's all that came in. If you have any other questions, obviously Wayne mentioned, I don't know if you want to back it up maybe one more time to that slide with the contact info, just in case anyone still needed to jot that down there. Otherwise, I think we'll go ahead and wrap up. So I just want to say thanks again for everyone joining and for all those good questions. And then just one more reminder that the presentation and the audio will be available at no-tillfarmer.com. And then you'll also get that in your email, usually, like I said, within 48 hours or so of the conclusion today. So one more time on behalf of Cultivates and No-Till Farmer, thanks for joining us today. And if you have any other feedback for us on other presentations you'd like to see, you can always go to No-Till Farmer and let us know there as well. Wayne, any closing comments?
Nope, just thanks for everybody for joining in, and hopefully you'll take a shot and take a look at an acetate instead of an EDTA. That's what we're here for.
All right, thanks everyone. Have a great rest of your day.